Okay. So here we are. Last lecture for exam 2, lecture 17. And from your syllabus, this is the functional morphology and locomotion and feeding of fishes. So we're going to look at how fishes are designed to swim and also how they're designed to capture food items. And so what we're going to do is note some diversity here and also differences between different taxonomic groups of fishes broadly. And so to begin with, I'd like to just say that, you know, we've explained that even through the taxonomy portion and moving into then the systems portion of this class, there's a lot of variation among fish or fishes. And whenever I say there's a rule, there's always an exception to that rule. And so when we talk about this, we're really talking about some general trends. Like, so for instance, more advanced fishes generally have a trend of losing the heavy armor, for instance, that the ancient fishes like the placoderms had. In reducing their armor, they become lighter and more agile. And so a typical fish, for instance, might be shaped like this, what we call a fusiform. This is a designed to be a swimming machine. But we also see this form here showing up over and over again, and it seems to not follow our phylogenetic distribution, which means that the eel-like shaped body is obviously successful as well. It's evolved several times in marine fishes as well as freshwater fishes. And that the most advanced fishes have a kind of odd shaped body. You know, when we look at things like the puffers or, you know, this ocean sunfish, mola mola, has a very ridiculous body shape. And so oftentimes we have exceptions to this general trend of being designed to be agile and fast swimming. So lots of variation here. And again, as I opened up in the beginning of this course, fishes have a very complicated phylogenetic history, meaning that you see lots of examples of convergent evolution where the same structures or forms are reinvented or resurrected. And that's okay, that's fine. It shows that, for instance, the hagfish was doing this first, and then the eels said, well, you know what, that works pretty good, let's do it like that. And then the freshwater catfishes like the electric eel said, you know what, that works pretty good, we're gonna do it again. And it happens over and over again. Likewise, the majority of fishes in existence have this more general fish body shape, which indicates that it also is a pretty successful strategy, because if you looked at the majority of fishes, they actually have this body shape. A handful of fishes, like the flatfishes, have said this is kind of a successful body form. And, you know, if we have to migrate both of our eyes onto one side of our head, we can do that. And we can lay on the bottom and hide and then wait for stuff to swim by and eat it. And so that works for some. Also, you know, a lot of things like the pupaid fishes, the herrings, and the shads, they have what we call a keeled body, where they have a very narrow ventral aspect to the point where it's almost razor-like, very thin. And this allows them to have camouflage when other fishes are viewing them from below. Imagine if this flounder is looking straight up, there's not really a whole lot to see on this very narrow aspect of the body. And so a lot of times they just see right by it, and this fish appears completely camouflaged. So the structure there reflects some kind of important life history trait. And likewise then, some fishes have adapted completely different body conformations. And so we're gonna look at how these different bodies, these forms all supply some kind of niche or function, and we can infer a lot about the biology, the feeding biology, as well as the locomotion, simply by just looking at the fish and understanding principles of physics. So for instance, in this case, it's the principles of light waves moving through the water column to know that this is a good form of camouflage. And so with that, I just wanted to illustrate lots of different body types, lots of different forms, and really we can infer function from form, and that the forms are not fixed in fishes. They're very plastic. And so you can have fishes even from very closely related families that have quite different body shapes. And that shows you also when we talk about the rapid evolution of fishes, why that's important, is that these things can change quite quickly. So the first thing I want to look at then is just outright locomotion. All right, so how a fish moves through the water. And again, going back to the fact that water is quite a bit more dense than air, these things, in order to move through water, they have to deal with this density. And so we have to look at streamlining a fish to reduce drag resistance, for instance. And so in some cases, fish want to reduce their drag resistance so they can swim as fast as they can. And other times, fishes actually might want to increase their drag resistance because if, for instance, if they're swimming around coral reefs or in tidal areas, they might want to actually be able to go with the flow, so to speak, it sort of helps them from being tumbled around. And so that's why you sort of see some really odd shaped fishes in these areas. They almost want to have a little bit of drag resistance. And so body parts of the fins providing propulsion is indicated here in the crossed hatch of these figures. And so what parts of the fishes allow for the locomotion? So in the case of anguillid eels, and again, this is named after family Anguillidae, the true eels, but like I said early on, you could have easily called this mixiniform swimming because evolutionarily the hagfish came first, but we just don't give the props to the hagfish, unfortunately. So most of the body is used in swimming for the anguillid eels. And that basically only the head isn't. And remember, a lot of anguillid eels might not even have pectoral fins. And so these things literally just swim through the water like a snake. Now, evolutionarily, this is a very successful form of swimming because a lot of fishes have adopted it. Even after it's been lost, it comes around again. And also, larval fishes oftentimes lack fins. And so when they're first developing, after they've hatched out of an egg, most larval fish actually use anguilliform swimming. And so here's a video of this with a moray eel I'm going to show, but note that the majority of the body is used in this propulsion. Now, as successful as this might be, because a lot of the body is used, it also indicates you're moving a lot of muscles. So this might be quite bioenergetically demanding when you think about it, because you're using a lot of your body to generate your forward locomotion. And so that means you're contracting muscles from your head to your toes, so to speak. Not that fish have toes, but it's sort of, the analogy sort of, I guess, does it justice. But so watch this watch this moray eel swim, and you'll see what I'm talking about. It has to use its entire body for locomotion. So if you notice there, the eel sort of generates a kind of an undulation or a sinusoidal wave originating right behind the head that then propagates all the way to the tail. And so it sort of swims through the water like that. But it's using the majority of its body to generate this propulsion force. Now, this figure here sort of shows the different swimming types. And so we call this is trunk and tail. Really, I'd almost say almost all the body. Now we look at what we call subcarangiform, carangiform, or thunniform swimming. And again, these are sort of named after the families, right, of fishes. And so these are things such as salmon, jacks, mako sharks, and tunas. You know, they might use, for instance, half of the body. Sometimes it's the caudal peduncle and the tail. But notice that, you know, most fishes are sort of a generalist, and so they have this what we call subcarangiform swimming, whereas the thunniforms use primarily the caudal peduncle and the caudal fin. These are more of a fast swimmer, because as much as using a lot of your body might help you generate a propulsive force, it also generates a lot of drag. And so the least amount of drag is generated by only moving your caudal peduncle and your caudal fin, which is why then this form of swimming is faster in the water column. This is more of a generalist fish swimming. So here's a video there showing this subcarangiform swimming, and note then the portion of the trunk of the body and the caudal fin being used. It's not going to be nearly as extensive as this previous video of the moray eel. So here are some tunas, and note then, primarily when these things are swimming, you'll be looking right here at the caudal peduncle back to the lunate caudal tail, or the caudal fin. This is where the majority, the fish is moving this part of its body. As these fish swim by, that's pretty much the only part of their body they're moving. They're just sort of swinging their tail a little bit. And they're almost using their pectoral fins really to steer. But generally, the majority of their forward locomotion force is generated just by that latter half, or latter third, if you will, of the trunk of their body. So that's where they generate their majority of their propulsion force. Now, when we talk about astraciform swimming, these are things like the boxfish. These things are kind of oddly shaped bodies. And note, we're going to watch primarily just the caudal fin and also the pectoral fin. Really, I think the pectoral fin should probably be a little bit cross-hatched on this one, because these two fins then are oftentimes what generates a lot of the forward propulsion force. So astraciform, we're going to show a puffer fish here, or a boxfish rather, swimming up towards the camera, and you'll be able to see that. So different parts of the body are being used to produce this forward locomotion. And so this boxfish is out here. And you can see these more generalist fishes up here showing this carangiform swimming, or subcarangiform swimming. But note, these fishes here are using sort of half their trunk of their body. Look at this thing here. It's primarily using, look at that, pectoral fins and that caudal tail. And then, of course, then sculling activity of the dorsal fin and the anal fin. So you can sort of see those fish then side by side and see different parts of their bodies are being used to create locomotion. So again, all fish swimming in the water, they don't always do it the same way. And so also then when we look at the tetraodontiform, ballistiform, again, family Ballistidae, the trigger fishes, and then diodontiform is the puffers, very similar here to the astraciform swimming. You're going to see a lot of sculling activity here with the dorsal fin and the anal fin, but unlike this ostraciform swimming, I think the example I've given, I'm going to show you here as a triggerfish, you're going to see even less use of the caudal fin itself proper. The caudal fin almost acts like a keel to help steer. It's primarily going to be sculling activity, the dorsal fin and the anal fin. So here's a triggerfish swimming. And note that really you're looking at the dorsal fin and the anal fin moving. And notice there's not a whole lot of caudal fin movement. They use the caudal fin a little bit to steer along with their pectoral fins, but most of that forward locomotion is generated by the sculling activity of the dorsal and the anal fins. So kind of cool. Different parts of the body. Now, when we look at, you know, when we talk about undulation of fins, you know, we've got the radiiforms, which sort of use their pectoral fins. Really, I mean, the stingray, the rays have a ray-like body, so they're kind of unusual. I just would group them in their own category. But, and so for the sake here, any of this is undulation of fins. The bowfin undulates its dorsal fin, and in doing so, it has both forward and also reverse locomotion. It can swim forward and backward by doing this. And the knife fishes can also do this, only instead of undulating their dorsal fin, they undulate their anal fin. So I'm going to show you here a video of a bowfin doing this. And so watch the dorsal fin of the bowfin, and you can see this is how it locomotes. So here's the bowfin right here.
This place is fabulous.
Doesn't it look all pretty in there? Oh, look how beautiful that is. Yeah. What's beautiful? Wow. No pictures. No pictures.
Hold on, sweetie.
We're gonna make some pennies and then we're gonna do a nightly change, okay?
You see that when the bowfin turned around there to swim away from the gar, you can see it fluttering its dorsal fin. All right, and then it uses its pectoral fins kind of like paddles to help steer. Same with this rounded caudal fin. And then last but not least, we have the labriform swimming. Family Labridae, the wrasses, and this is primarily flapping its pectoral fins like wings. So here is a video there of the wrasse labriform swimming. And pay attention primarily to these pectoral fins flapping. And you can sort of see that very little of the rest of the body of that fish is actually moving. It's literally just flapping its fins like wings. So lots of different ways that fish swim. And again, you can sort of look at their forms and see then how they fit into these categories of swimming, just by looking at the structures of how the fins are placed and also the shape of the body. So this part of this slide here then just goes over those exact things we talked about in the videos that I showed. So anguilliform swimming is seen in eels, dogfishes, other elongated sharks. All but the head contributes to the propulsive force. And then fish larvae swim in this way as well, since in many cases the fins are not formed. Subchorangiform swimming are things like the salmons and cods. They use the posterior half of their body. Chorangiform swimming are things like the jacks and the herrings. They use the posterior third. And then thuniform swimming are the tunas and mako sharks, use mostly the caudal peduncle and tail, the caudal fin. Ostraciiform swimming are the boxfishes. They use the caudal peduncle and caudal fin. And then tetraodontiform, balistiform, diodontiform, the triggerfishes, the ocean sunfishes, and the pufferfishes use these oddly shaped dorsal and anal fins to undulate, flutter, in a form that in a manner that we call sculling. They swim kind of like a helicopter. They use this kind of swimming. Ragiforms, again, undulate their pectoral fins. Ameiforms undulate the dorsal fin. And then gymnotiforms are like things like the electric eel, which again is more closely related to a catfish. They undulate their anal fin. Labriform swimmers use the pectorals for slow swimming, and then subchorangiform for fast swimming. And say the majority of fishes, right, 30,000 species, they're going to be a generalist. Oftentimes a bluegill, for instance, you're going to see it might use its pectoral fins to sort of paddle against the water when it wants to turn to the left or turn to the right. And then if it wants to swim away from a predator like a largemouth bass, it will use its trunk, the posterior third of its body, and the caudal fin in chorangiform swimming. So most fishes are generalists. They'll combine chorangiform and labriform swimming. Labriform for slow speed, chorangiform for more faster locomotion. That's what most fishes do. So despite these names here, they're sort of named after the families that use them almost exclusively, but most fishes, again, are generalists. When you look then at the proportion of the body that is used in locomotion, this figure here shows in the shading here what proportion of the body is used for locomotion, and to say that increasing proportion of the body undulates versus less in the thunniform. And when you look at this then in regards to amount of head movement, so in order to generate this undulation down the side of their body, if you're going to propagate a wave down the majority length of your body, that means you got to start in the head, and that means the head's got to swing pretty extensively from one direction to the other as you're moving through the water column. And so head movement is maximized here. Now, if your idea is going straight ahead and being able to see straight ahead, number one, for predators, but also to identify prey items, you sort of want to keep on target, stay on target, right? So oftentimes undulating with a lower proportion of your body allows your head to swing less. So these things can actually then swim more effectively straight as a vector through the water column, which is part of the reason why also they can swim a lot faster, because they're not, this left, right, left, right, left, right, there's a lot of energy being used here, right? On a left, right vector that's not helping you move forward through the water. Whereas a lot less energy is being used left, right, left here, and more energy can then be dedicated towards moving straight ahead in the water column. But again, most fishes sort of are somewhere here in the middle, right? They sort of sacrifice a little bit of this to combine it with a little bit of that, okay? Now, ragiforms, of course, the skates and the rays are totally on their own here because, I mean, this is quite a unique thing. What they do is they propagate a wave, an undulation, or sinusoidal activity of the pectoral fin that starts here by the head and then propagates posteriorly. And in doing so, they sort of flutter their fins, kind of like a bird flies. And so here we're going to show a video there of the ragiform swimming, which again, I mean, it's ragiform swimming. I mean, there's no other way to put it. It's how stingrays swim. Very So that is ragiform swimming. And so they are unique in their swimming. And so again, adding even more to the diversity of forms and body function of locomotion, lots of different ways to move through the water column. And so these things are oftentimes named then after the major families which use these different forms of locomotion. So lots of different ways to do the same thing, which is move through the water. When we look then at the structure of the fish on the inside, sort of knowing that most fishes, again, are generalists. They use a majority of their trunk. For instance, again, here's a generalist salmonid, soft rayed fin here with an adipose fin. I'd go ahead and call that family Salmonidae. This is an example there. We've got these bundles of muscle fibers sort of organized like these W's, right? It's kind of like a W. And these things are called myomeres. All right, here is sort of these cross-hatch patterns here. They're sort of bundles of muscle fibers arranged like this. And these bundles of muscle fibers are covered by a myoseptum, so they're kind of separate. What happens is when this fish wants to undulate, what happens is all these muscle fibers in that little W pattern contract. And normally when I've got students in the classroom, I've got little models I can show this, but what that does is that means the fish is going to sort of bend. It's going to bend. And so that means that it's going to propagate a wave where this W contracts, the fish bends a little bit, and then the next W contracts, and then so on and so forth down. So what happens is this sort of bending wave propagates down the side of the fish, which then creates this undulation. Because as the left side of the fish, as the left side of the fish contracts, then the right side of the fish will contract right behind it. And so it sort of then gives this sort of propagating S-like wave down the side of it. When we look at the cross section of the fish, so if we cut it like this, we can see then that these myomere bundles sort of appear circular. And so when you look at the side of the fish, this is your W's are like this, but when you look at the cross section like that, they sort of are organized in these like sort of ring-like patterns. So when you look at a fillet here, you can see these ring-like patterns here on the salmon fillet here, but on the side of it, you can sort of see it's got this W or V-shaped pattern. And that's sort of these organizations, these myomers. So you can look at a fish fillet and actually see these structures. And these myomeres, when I say that they're sort of surrounded by a myoseptum of connective tissue, you can see the muscle fibers here are the orange colored parts, and the white is this connective tissue then that surrounds them. It sort of bundles them together. And so this myoseptum is made up of collagen and some kind of gel matrix. And you can sort of see it then here in this fish fillet. And so this is, again, when we talk about fish, remember, we're talking generally. Most fishes of your 30,000 species, they're going to swim like this. This is what their muscular structure looks like. And also, this is why the majority of those fishes, they're good eating fishes, because notice how much of this body, when we say we've gotten rid of our heavy armor, we've made a very light skeleton that allows us to have fast locomotion relative to our size. All of this is edible. That's why fishes are a very good source of food, because the vast majority of them are edible protein. Very little here is bony elements. And there's only like a very light scale of armor on the outside. So most of the components of the fish are edible. Hence the majority of the mass of the fish is consumed. When we look at caudal fin development, there's also some specializations here. So looking at the shape of the caudal fin, or the tail, can give us an indication of swimming specialization. So in addition to which part of is it the trunk of the fish that generates the wave, or is it just the caudal fin? We can just look at the shape of the caudal fin, and it tells us a lot about this. So for instance, fishes that have a rounded caudal fin. I like to look at this figure like this. From top down, a rounded caudal fin, high, maneuverability, high agility, high drag resistance, low speed. Down here, we have low agility, low maneuverability, high speed. So there's a trade-off. These clownfish, for instance, have a rounded caudal fin. You've seen the videos of the clownfish swimming around inside of the sea anemone, taking care of it. These things can spin around, swim in all different degrees, 360 degrees around them, very rapidly. They're very agile. They can swim all around inside the tentacles of a sea anemone, no problem. A tuna? Oh boy, these things are swimming so fast that it's like a jet airplane. If they want to turn around, they got to swim and come around in a circle to go back. You know, we showed videos of things like thresher sharks and tunas and billfish swimming. If they swim past their target, they got to swim all the way around in a circle and come back. Very low agility. They're very fast, but they almost have to turn, you know, in order to turn around, they got to swim forward and come back around for a second pass. So again, very fast, maybe not so agile. And then all these other different tail forms are somewhere in between. So again, the truncate tail is going to have maybe a little bit less agility, a little bit more speed compared to a rounded caudal fin. And then the emarginated caudal fins maybe got a little bit of a segment cut out here, a little bit more speed, a little bit less agility. The forked fins are maybe even a little bit more so. This is a little bit faster swimming fish, maybe a little bit less agile. And then this here is the extreme. And the reason why the drag resistance is important is that this is a paddle, like an oar that you use to paddle a boat. The more area you have in two-dimensional space here, the more water you can push with that paddle. But the more water you can push, the more drag resistance by definition it creates. Whereas if you take that and punch a hole out of it, this thing creates less drag resistance. And so as water flows by it, it's going to create less resistant pull. And so that means that this fish can move through the water column more quickly than this fish here. But this fish can stop because that tail has got more drag resistance. It can put it out and stop at a very quick time frame and then turn to the left, for instance. Whereas this fish here cannot do that. These can put the brakes on fast. These guys don't have the tail form to put the brakes on. But the trade-off is they don't put the brakes on. They swim right through it and come back around. So that's sort of the trade-off there. Look at physics. So punching out this hole in the tail reduces the drag resistance. But what it means then is the trade-off is they cannot stop and turn around so quickly. These guys can. But again, as usual, most fish are somewhere in the middle. We are what we call generalists. All right. So high maneuverability, low speed, low maneuverability, high speed, and then everywhere in between. As you might expect, 30,000 different species of these things. Now, we talk about, again, the evolution of fishes. We started off as these heavily armored things tumbling around in the muck on the bottom. Hey, we don't need to swim. We're just going to hang around on the bottom eating the muck. As long as we have armor, nothing can mess with us. Well, the problem is evolutionarily, other stuff started evolving jaws, which means that, in fact, yes, other stuff can now start messing with you. So what do? Get rid of the armor. Let's go ahead and say we can swim. Let's reduce our density so we can swim fast. So there's two ways we did this. The first is what we call dynamic lift, and the second is what we call static lift. Now, when we look at things like elasmobranchs or chondrichthyan fishes, dynamic lift is the generation of lift by appropriately shaped, angled, and positioned fins. So if you look at the structure of this fish here, I've drawn it. I could have easily, I drew a shark here, a chondrichthyan fish, but I could have easily drawn a sturgeon for instance, because sturgeons have the same kind of thing. Heterocircle tail and these kind of planing, what we call planing pectoral fins. Also, I could have drawn a paddlefish. Paddlefish have a heterocircle tail, and they also have planing pectorals. They're bony, they're chondrostii. This thing looks just like a jet airplane. It literally looks, these fins are designed just like the wings of an airplane, and this caudal fin is designed just like the tail of an airplane. And really, all that we need to do is have the trunk of the fish provide some kind of undulation force so it swims straight through the water column. The tails and fins provide downward force, which means then that the fish, as it swims through the water column, actually swims like this. It's got an upward vector to its motion. So as long as this thing is swimming forward, it's not going to sink to the bottom because of the way it's designed. This is called dynamic lift. Now, the problem is in order to use dynamic lift is you have to constantly be swimming forward. If you stop swimming, you will sink. Okay. So if you ever want to rest and stop swimming, just like an airplane, once you get to cruising altitude, if the pilot shut off the engines, the plane's coming down. Okay. So the other way we can deal with this then is with what we call static lift. And static lift is, what if we reduce our specific gravity in the body? So again, get rid of the armor. We don't need heavy bony plates. Let's reduce our dense body parts. Let's, in the case of chondrichthyes, let's get rid of the skeleton entirely. We don't need it anymore. It's not important. Let's reduce our skeleton, get rid of it. That way we can replace it then with muscle that we can use then for this forward locomotion. Also, let's include some low density materials. What's less dense than water? Gas. Okay, we can use gas. Oil. All right, we can use oil. And then in the case of chondrichthyes, cartilage. Cartilage is a lot less dense than bone. So let's go ahead and replace our skeleton with bone, or with cartilage. So sharks use dynamic lift, continuous swimming, to maintain position in the water column. They also use squalene. Here's the structure of squalene. It's a lipid. It's produced in the liver and stored there. And so this lipid then reduces the density of the shark body, giving it static lift. And so most fishes actually use kind of a combination of this. All right, so we've gotten rid of our cartilage. We're using dynamic lift with our fins. And we're going to store oil in our liver to reduce our specific gravity so we don't sink to the bottom like a rock or a fish without static or dynamic lift. Okay, so chondrichthyes do this. Now, bony fishes, teleosts, primarily will use static lift. And because they primarily use static lift, you now decoupled our forward momentum vector of movement from our upward lift vector, which means we don't have to just rely on our fins and their placement to maintain our position in the water column. We'll use mostly static lift in the more derived teleost fishes. So the first form of this is what we call buoyancy control via a physostomous swim bladder. Now, I said earlier in the lectures that this is the taxonomic characteristic that less derived bony fishes have what are called a physostomous swim bladder. That is a swim bladder that is connected to the esophagus. Here's our mouth over here. Water comes in through the esophagus through some pneumatic duct into the swim bladder. And then this is to our gut. This is to our stomach. So we've got a sphincter here that can control releasing air out of this. So what happens is that if the fish wants to become more buoyant, it swims to the surface, gulps water or gulps air. That air comes in here into the swim bladder, and it squeezes that duct shut and holds that air in here. Because it's holding air in the swim bladder, it has just reduced its overall specific gravity. Now, if the fish wants to descend and increase its specific gravity a little bit, all it's got to do is release this sphincter and belch a little bit of air out. And then it can actually descend. So this is a common and primitive soft ray teleosts, or less derived, things like herrings, Clupeidae, and salmonids. And again, it's just filled with air that the fish gulps at the surface. During development, ontogeny is development. During development, all swim bladders start off as a physostomous swim bladder. That means that even if a fish has the other type of swim bladder, the physoclystis swim bladder, which is on the next slide, they all start off like this. And then during development, this duct closes off permanently. And the fish are no longer able to do this. Only as larvae or really small juveniles can they do this in the physoclystis fishes. But in the case of herrings and salmons, the adults, they do this throughout their entire life. They have what's called a physostomous swim bladder. Now, as you might imagine, a fish living on the bottom of the ocean is not going to swim all the way up to the surface just to gulp air. That's ridiculous. Why would you do that? And so what they've done is they've evolved what's called a gas gland. And so again, notice here's our physoclystis swim bladder. There is no duct connecting to the esophagus. I don't even show it. No duct. It's what we call a closed swim bladder. There is this gas gland that literally secretes oxygen gas in here. This swim bladder is filled with molecular oxygen that's been extracted from the blood. Now, if you recall, when I talked about respiration, I talked to you about this root and bore effect. Well, what it does is the gas gland is essentially an acid pump that creates a super root and bore effect, which forces the offloading of oxygen in. And then if it wants, the fish can then inflate it here. And if it wants to deflate it, all it does is allows the oxygen to diffuse out and back into the blood. And then it goes about its merry way. Now, the problem is that fish cannot do this rapidly. So if you take like this here, this grouper or something, if you take a fish like a physoclystis fish and you catch it at the deep sea and you reel it in and pull it all the way up, look at this. Boop. As the pressure changes, the fish is not able to void gas out of this thing very rapidly. And so what happens is as a fish gets into less deep water, that swim bladder expands rapidly and can literally push all the guts out of the fish, out of the mouth of the fish. And so physoclystis swim bladders are often found in moor-derived fishes, such as rockfish, cods, and basses, and also fishes that live in the deep sea. Ones, again, that are not going to swim all the way to the surface just to fill a swim bladder. Now, fishes that live in the really deep ocean, because gas is compressible, they might actually fill the swim bladder not with oxygen, but with things like oils or fat. Because like water, oils and fat are really incompressible. And so they don't have to worry about this pressure differentials based upon depth. Lipid is still going to give the same amount of buoyancy at depth, and it doesn't compress. Whereas gas compresses at depth. If you go all the way 6,000 feet down with a balloon that's this big, by the time you get down to 6,000 feet in depth, that little balloon is this big. It gets compressed. Likewise, if you start off with something this big around at depth, 6,000 feet, and you bring it all the way to the surface, boom, that balloon is now huge to the point where it can pop or at least force all the guts out of your mouth, which is not that good. So this is a consideration. You know, a lot of people look at like catching fish at depth and bringing them up. And this is a handout from Florida Sea Grant talking about here's a swim bladder of a fish, and if you catch one, if you bring it to the surface, that swim bladder will increase its volume because of the pressure change, and it can evert the stomach, for instance. Well, you can actually take a needle and you can pop it. And you know what they do is then you say that once you pop it, then the stomach deflates, and if you release that fish, then the fish might then have a way to potentially recover from this. So venting the gas helps the fish survive potentially. So this is one of several handouts that talk about this, but this is only for physoclystis fish. A physostomous fish doesn't have to worry about this, because if you were to rapidly bring it to the surface, all it does is it burps and all the air comes out. Only the physoclystis swim bladers who don't have this esophageal connection have to worry about this. So that's one handout there. When you look at position of structures on the fish, let's look at the mouth. This is also part of our moving into the feeding apparatus now. If you look at the position of the mouth, if mouth is superior, then we can sort of conclude that things like the half beaks, remember, related to the flying fishes, family Excelsidae, the hatchet fishes, they have superior mouths. These things, you can tell, probably live at the surface, probably feed off the surface. So you can sort of tell us a little bit about their ecological niche. Most fishes, of course, are generalists. So they're going to have a terminal mouth, which means they can feed on stuff in the water column in front of their heads. They can also feed above or below. Terminal mouth. You know, these things can feed on other fishes. They can feed on zooplankton. And so they're a generalist. They can feed on a lot of different stuff. And then you've got certain fishes, for instance, the suckers, family Catastomidae. And I said that generally speaking, you know, the fish mouth sort of tells you, the jaw structure sort of tells you whether it's an advanced fish or a less derived fish. But in some cases, like in the case of the suckers, they're an advanced fish, but they have this underturned mouth. But really, this is simply an adaptation for feeding off the benthos. So they feed off the substrate. So you can sort of tell, hey, these fish probably feed off the benthos. So you can look at how the mouth of the fish is designed and infer then likely what it's eating, what kind of role it has. We talked about the diversity of family Cichlidae, one of the most diverse families of fishes in the world, family Cichlidae. I talked about the cichlids of Lake Malawi. These are the Darwinian finches. And I talked about, as much as Darwin showed variation in the beaks of birds, of finches that were all closely related, we see a similar phenomenon. Different features of the mouths of very closely related family of fishes. And in some cases, for instance, like the Mozambique tilapia, they might be designed to feed on vertical substrates, like to scrape algae off of stones or rocks. Some mouths are designed to scrape in a downward angle, more designed to, for instance, feed off of the benthos. Some are designed to manipulate things such as insects, so insectivorous things. Some are designed to bite, so taking off pieces of other fish or fins. Some are designed to feed at the surface, a superior mouth. Some are designed to feed and forage in the benthos, not just to scrape stuff off of stones, but maybe more of a pipette mouth, designed to sift through the benthos to find invertebrates, for instance. Some are designed with this suctorial pipette feeding, this predatory quick motion to inhale something whole, whether that be small things like invertebrates or larger prey items like other fishes. Lots of variation here in the feeding structures of family Cichlidae. And oftentimes then this includes variation in what we call mandibular or oral teeth. We talked then about how some fishes, for instance, eat vegetables. Like the gars have these long, sharp, pointy teeth to help them consume vegetation. Don't write that down. These things are piscivores. They're using these teeth to help trap prey that swims, that's a live animal. And that these teeth then are designed to trap that and prevent it from escaping. The bowfin, here's a bowfin skull. Here's a great white designed to bite chunks out of something. Here's a piranha designed to bite chunks out of something. Here's a pike, again, designed more like the beak here of a gar. It's designed to bite onto something and hold on to it so that it doesn't escape, and then it will slowly work that thing into its throat to swallow it whole. Sometimes they're swallowing stuff whole or tearing stuff. Other times they're biting chunks apart. But these are the oral teeth, all also have different mechanisms. And then here again is this most evolved suctorial feeding jaw, something you might see in a largemouth bass. That as this mouth opens, the premaxilla swings forward, or the maxilla swings forward when the mandible depresses, and this premaxilla then swings forward, it greatly expanding this buccal cavity, which causes a tremendous negative pressure that allows a fish to suck something in whole. So when I talk about trapping something and slowly working it down the gullet, here's a video. Pike versus duck. And so watch this. So the jaws are designed to hold on to that bird until it finally becomes tired or cannot fight.
Nee, die vermoorden elkaar. Jezus, hij zit diep in zijn keel.
So the bird is trying to peck. I believe that's it. The bird never resurfaces, and that pike eats it. So again, the jaws are designed to hold on to a prey item to prevent escape, and then eventually it is swallowed. So that is different types of jaw structures there. And so again, the capturing prey or inhaling it straight, lots of different strategies. We talked about the different jaws. We have an oral jaw and also a pharyngeal jaw. And this is, the oral jaw is primarily designed to capture our prey item. Make sure it doesn't get away. How do we acquire it? But as fishes evolved, the more advanced fishes have a very sophisticated pharyngeal jaw. This is, again, inside of the skull, designed to pulverize stuff. So the more advanced fishes have a very sophisticated jaw here, crushing. And look at, this is what these pharyngeal teeth look like. There's an upper tooth and a lower tooth, and they look just like the molars in your jaw. And that's exactly what they're designed to do. If this is designed to cut, shred, or hold on to something, this is designed to pulverize it, whether it's a shellfish or whether it's a nice fleshy thing that it wants to pulverize to kill it before it gets into the stomach. Either way, that's what this is designed to do. And as fishes evolved, this became much more sophisticated. When we dissect something, this is a bony fish. I can tell because it has, these are our gill arches. 1, 2, 3, 4. 4 gill arches, bony fish. Here's our esophagus. So this is our coming down the gullet. And so when we look here, this T here is our tooth, our bony tooth pads. On the upper and lower. These T's here are pharyngeal teeth. And this literally, the gill arches are flexible. This literally can pulverize like this. The fish controls this. They actually can chomp with this. And again, like I told you, how the gill rakers, if the filaments of the gill are involved in gas exchange, the gill rakers are part of this feeding apparatus. So the function of the pharyngeal teeth then is to grind food particles, which is analogous to our molar teeth. And the fish literally can chomp. This is a jaw here that it can control independent. It can close its oral jaw, and it can close its pharyngeal jaw. It can close either one. Close them both, open one, close the other, however they want to do. So right? That's the fish jaws. We look then at our gill rakers. Again, they're angled sort of forward here. Kind of like if you're going down the throat of this fish, these are like hard, heavy, bony spears pointing you right in the face. And the fish actually can control these. So, for instance, the gill rakers are bony extensions of these gill arches, and they're involved. So as you're a prey item traveling down the throat of this fish, these things can basically form a cage and prevent you from swimming out. The fish can control this. It can angle its gill arches to sort of make a bony cage. So again, we can look at the features of these gill rakers. And number one, they're taxonomic characters. We can tell the difference between species just by looking at the number of these things. You know, you can count them, for instance. Also, just looking at how they're structured. So a piscivore is going to have very sharp, heavy, pointy, spiky gill rakers. And again, this is designed to keep living fishes from swimming out. Remember that most prey items are being swallowed alive until they get to the pharyngeal teeth that pounds it to death. They're going through the oral jaw quite aware of where they're going. And so this here prevents them from swimming out. Now, most fishes, again, are omnivores, and so they're going to have some intermediate kind of gill raker structure. They're more of a generalist. And then there are planktivores, like here. Here's our paddlefish. They're going to have very fine, thin gill rakers, like a comb. And what happens is as water passes through the mouth of a paddlefish into the buccal cavity, into the opercular cavity, plankton will get caught on these fine gill rakers because they're coated with a mucus. And then once the gill rakers are coated, the fish will swallow that. So they can actually be used to filter feed. Again, I mean, it's no different than the ancient bryozoans were using these gills to feed and do gas exchange millions of years ago. So involved in feeding as well as, and this is the gas exchange side, this is the feeding side, so the gill rakers. So you might want to know the difference here and variation of different gill rakers based upon feeding strategy or trophic level. Piscivores, omnivores, and planktivores, right? And that they're involved in helping to trap different types of things that might be trying to escape the inevitable. Now, when we look at overall diet strategies of fishes, you know, there is a couple different key features here. Again, this is a lot of review from Intro to Biology. There are 3 primary types of fishes. There are herbivores. You know, usually limited to a range of plants. And again, they could be both marine, freshwater, estuary. There are carnivores. Carnivores includes fishes that eat other fishes, which we call a piscivore. There are fishes that eat other insects, or that eat insects. They're insectivores. And as I showed, there's a video of pikes that eat ducks. But you know what? A pike's going to eat anything. So I would call them an outright carnivore. They're not going to be specific in what other type of meat they're eating. And then there are omnivores. Often small invertebrates, mixed diet, minimum specialization. They eat pretty much anything. I sort of show here the picture of a common carp, because I mean the common carp is pretty much going to eat anything. And I'd like to just point out here that all of these different feeding strategies can be found within the same family of fishes. So when I gave you your family designations, I sort of said, generally speaking, this is what's going on. But as an example, family Cichlidae, you're going to see all these different feeding strategies represented within a single family. You're going to have herbivores, carnivores, and omnivores. So it is very highly adaptable, very easy to change. Once we designed this sophisticated jaw, all we have to do is make slight modifications to it to say, are we eating plants or are we eating animals, or shoot, let's just go ahead and eat both. Doesn't really matter. If it's got energy in it, let's eat it. So when we talk about dietary specialization, you got what we call euryphagus. There's many types of food, little specialization. Again, the common carp, it's going to eat pretty much anything. It's going to eat other fish. Cool. It's going to eat algae. If it needs the plankton feed, it can do that. It can eat detritus off the bottom. It can eat muck. You can feed it a cheeseburger. It'll eat that too. There are what we call stenophagus, which have a limited diet. For instance, only being a piscivore. It only eats other fish. That's kind of stenophagus. Or it only eats a certain type of plant. And then we have what's called monophagus. The prey is only maybe a single species, so very restricted. Now I'm going to tell you this. This is things like koala bears and panda bears. You know, this doesn't really happen a whole lot in fish. There's not a whole lot of, you know, maybe things like weird things like the inquilin pearl fishes that live inside of the anus of a sea cucumber that specialize on eating very specific things like the fecal material of a sea cucumber. This is kind of rare. Most fishes are what we call euryphagus, a little specialization, carnivores. I might extend this to saying even euryphagus omnivores, because they literally will eat anything. Even an herbivore or a carnivore isn't really going to reject eating an edible if they can get energy out of it. So like a grass carp, for instance, is typically an herbivore. But if a fish had spawned adhesive eggs that are sticking to the seaweed that the grass carp is eating, it's not like the grass carp's going to go, oh no, I'm not eating these eggs. I'm spitting them out. It's just going to eat it. And so they're not really, there's no specialization. They're just going to go ahead and eat it. So they're what we call opportunistic, in the sense that even if we have an herbivore that has a small mouth that's designed to eat vegetation, if a little fish swims by and, hey, I can eat that, it's going to go ahead and eat that. And so they're opportunistic. And so I'd say euryphagus, little specialization, carnivores, and in some cases, I would even say euryphagus omnivores, because I mean, they pretty much will eat whatever they can. Now, you know, you always hear these stories about the stuff they pull out of the stomachs of sharks, like license plates and all sorts of stuff. And so, I mean, they'll try to eat whatever. And so they're very opportunistic. And so with that, I've got one more slide here on some interesting digestive adaptations, and then I've got a story. So here is a spiral valve. Now, I told you in the taxonomy portion of the lecture that certain fish species had spiral valves. For instance, a lot of the chondrichthyan fishes have these. Also, some of the less derived ray finned fishes have these. For instance, the chondrostii, things like the sturgeons of family Assapenceridae, also the paddlefish Polyodontidae. But generally speaking, I said that if a fish has one of these spiral valves, it's considered a less derived feature. And that is true. It's a feature of less derived fishes. So chondrichthyes, sturgeons, paddlefishes, the coelacanth, Latimeridae, and the lungfishes. It's kind of a throwback. And what this spiral valve does, this is sort of what a cross section of it looks like, is it increases the surface area and retention time for absorption of nutrients in the gut. So what happens is that, let's say I'm digesting something and I've got it coming through my gut. And so one, for instance, is this spiral or corkscrew shaped spiral valve. So I got stuff coming in and it's got to then spiral around this way and then this way and then this way and then this way. And it's got to go all the way down here, and see how long this is taking to go down these spirals, as opposed to just saying, yep, I'm just going to go right through here. And so it increases the gut retention time, which then allows more time for a digestive breakdown, but also absorption of nutrients. And so in this case, you know, sharks are oftentimes carnivorous. They eat other stuff that is living animals swimming away. And so sometimes this allows them then a more time to digest and absorb stuff. But digesting protein is really pretty simple. It's really pretty easy to break down protein. Oftentimes when we see things like sturgeons, who oftentimes can eat stuff off the bottom, the muck, so to speak, detritus, it takes a lot of energy to break down detritus to get the energy out of it. And so sometimes increasing this retention time allows for a more complete digestion of detritus, and also then a more complete absorption of detritus. Paddlefish are planktivores, and so sometimes, again, breaking down the plankton and increasing the absorption time. There's not a whole, you have to, basically, plankton is one of those things that if you're going to be committed to eating it, boy, you're pretty much constantly feeding. There's no time that you're not eating. And so oftentimes it's a continuous activity. And so again, making sure you extract the maximum amount of nutrients out of that diet. So there's a few different types of spiral valves here. Some are what we call like this corkscrew shaped spiral valve. Other ones are more scroll-like. If I took a sheet of paper and sort of rolled it up, and you sort of see this scroll-like shape, and some are just sort of rings. There are a series of plates, for instance, where there's just sort of invaginations in the side. These pockets, if you will. And some of these are found in different taxa, but I'm just illustrating this really. The take-home message here is that it increases time for digestion and absorption, and it is a taxonomic characteristic, mostly found in less derived fishes, but it can also be a taxonomic characteristic. For instance, these plate-like or ring-like spiral valves are mostly found in the lamnid sharks, as opposed to, say, Carcharinidae, which have a scroll-like spiral valve, which is different than family Squalidae, which might have more of a corkscrew-shaped spiral valve. And so to let you know that there's a lot of variation here, but really it's a design to help increase the digestion, retention time, and absorption time in the gut to maximize nutrient uptake. And generally speaking, the lower the trophic level of the fish, the longer the gut generally. So detritivores have the longest guts, herbivores, second longest guts, omnivores, the third longest guts, and generally speaking, piscivores or carnivores are very short guts like this. Esophagus, stomach, zigzag Z, spiral valve, and out. Not a very long coiled gut. And I sort of explained this then in the video of the dissection of the tilapia, noting that the tilapia is kind of an omnivore. It has a very long coiled gut. And that's different compared to, say, a piscicle, which has a short kind of like this. In some cases, like in the pikes, it's literally throat to stomach to anus. Like there's no little zigzag in it. It's literally the intestine's a straight shot. Because again, it's pretty easy to digest meat. So with that, here's a story. You learned it here at AEC 441. Coelacanths have fat heads. They're fat-headed fish. Now, that's not a poke at the fact that they're dim-witted or stupid. They're not. I mean, these things have been around for hundreds of millions of years. They're pretty cool. They do very well. But I told you that one of the ways that fishes regulate their buoyancy is by having structures in them that are less dense than water. And that provides static lift. And one of the ways they can provide static lift is, for instance, storing fat or lipid or gas. And so in the sense of sharks, they store lipid in the form of squalene in the liver. But in the form of bony fishes, teleosts, they can have this swim bladder, this air bladder that they can store gas in. Because air or molecular oxygen is less dense than water. But coelacanths live very deep, 2,300 feet below the surface. So again, they are not going to have a physostomous swim bladder that they're going to swim all the way to the surface just to fill with air. That's a waste of time. They have a single elongated swim bladder that they actually fill with fat. Now, they don't have a physoclystis swim bladder because I said that's a characteristic of advanced derived fishes, perciforms. The coelacanth is sarcopterygian. It's a lobefin fish. These things have been around before the physoclystis swim bladder existed. So they fill their lung with fat because they're at a very deep part of the ocean and gas is compressible, whereas lipid is not. And so if they fill their swim bladder with fat, it's not compressible at this depth. The skeleton includes a hollow tube of cartilage called a notochord that is also filled with oil. The spine is hollow. So this is actually where they get the name coelacanth. This means hollow bone or hollow spine or hollow back. The coelacanth's brain occupies 1.5% of the cranial cavity of the skull. And the rest of the skull is filled with fat, hence fat heads. Now, all these fats and oils and lipids provide coelacanths with the buoyancy they need in the deep waters of the Indian Ocean. In some cases, the Western Pacific. So the lipid here in the head and in the swim bladder provide them with buoyancy. And they actually continuously ooze a small amount of oil from their bodies. So unique characteristics of the coelacanth represent an early step in the evolution of fish to the terrestrial four-legged animals, which includes then the tetrapods, like and amnio amphibians. So the fat head of the coelacanth is a form of static lift. So with that, I'll now stop recording and I will entertain some questions. And it looks like we have a handful here of chats.